everybody, this is your Shruti ma'am and I'm your mentor at What Next Education. So today we're going to take a deeper look into enzyme kinetics. So remember that for your CSIR exam, this is a very important topic, and also for your DBT exam that is coming, this is super important. So definitely do take note of this and also start your preparation immediately. To help in your preparation, I have created AI tools and materials that can really support you in your preparation. And to know details about it, reach out to me on my Instagram page What Next Education, or you can also take a look at my website whatnexteducation.com. So this materials, these AI tools can exclusively help in your self-study preparation, and you will definitely be able to crack the exam in no time. Alright, so now when we look at enzyme kinetics, okay, let's first understand what an enzyme is. So an enzyme is nothing but a protein, okay, and this protein helps to accelerate the rate of a reaction, of any reaction, okay. So in our metabolic systems, all metabolic reactions require a catalyst in order to accelerate the rate of the reaction. Now how exactly it does this is because, now let's say I have a substrate in a certain potential energy, okay, at a certain level of energy I have my substrate. Now this substrate has to get converted to a protein, to a product, some product. Now if the product is at a lower energy level, I need to give out energy, so that becomes an exothermic process and I will get to the product. If the product is at a higher energy, I will have to take in more energy, so that becomes an endothermic reaction and I will take in some energy and reach the product. But in the process, I need to meet some transition state that are at an extremely high energy level. Okay, now in order to meet all of these people and then get to my product, it requires a lot of energy. And this energy is what I call as activation energy. Okay, now what this enzyme does is, it's going to lower this activation energy. So then what happens, I know I don't have to wait for so much of energy, only so much is enough for me. So when my activation energy is lowered, easily the rate of my reaction also increases. Okay, so that is exactly how the enzymes will bring the rate of the reaction to a much faster rate. Okay, but they do not alter any kind of equilibrium constant or the Gibbs free energy of this thing. So delta G, equilibrium constant, everything is going to remain the same. They do not affect anything, and in fact after the catalysis is over, my enzyme also remains as an intact product and it does not undergo any change as well. Okay, now when you look at an enzyme structurally, the enzyme requires a 3D conformation for its biological activity, and in the 3D conformation it will have a small pocket like structure called as the active site. Okay, and this is exactly where my substrate will come and bind to the enzyme for my process of catalysis to take place. But for the enzyme to function and to actually bring about the catalytic activity, along with the protein portion of it, which I call as an apoenzyme, I require some additional factors to help me, and these factors are called as my cofactors. Okay, now cofactors can be metal ions, okay, they can be a prostatic group, which is an organic group, okay, and it can also be a coenzyme also. Okay, anything, but this cofactor is definitely needed. Now this apoenzyme plus the cofactor forms what we call as a holoenzyme, and this is what is completely functional and ready for the process of catalysis. Understood? Now if you look at enzyme kinetics, okay, to understand how exactly this enzyme works, you need to understand a few other factors that play a role in the enzyme's functioning. So factors that affect the enzyme, okay, that affect the enzyme, okay, include your temperature and pH. That's the first thing, definitely. Okay, now if you look at temperature and pH, they all have an optimum temperature. at which the activity is maximum. Okay, now under that what happens is any region this side or that side, my activity will come down. So this is what I call as my optimum temperature or pH. Okay, that is very important to remember. Now when you look at the substrate concentration, what is going to happen here? When I increase my number of substrate molecules, my rate of the reaction also is going to increase. Okay, so this is my substrate and this is my velocity. Okay, now the rate of the reaction is also going to increase as I increase the substrate molecules. Why? Because if let's say you go for a haircut, okay, there are 3 stylists who are available. Then I have one person going in. Okay, one person will finish styling in 1 hour. 2 people will go in. 2 people will also finish in 1 hour. 3 people will go in. 3 people also will finish in 1 hour. But the 4th person has to wait in the line because all the 3 stylists are already busy with the 3 people. So same way in the enzyme also, the enzyme molecules have an active site where the substrate is going to bind and bring about my catalysis. In case my substrate molecule is done, okay, in case my enzyme molecules are already occupied, the next molecule has to wait in the line so that after that I can come and do my catalysis. Okay, so until a certain point my rate of the reaction will increase, but after a certain point it will stabilize. So this is what I call as my Vmax, or the maximum possible velocity that I can attain. Understood? Now for half the maximum velocity to attain, okay, half the value, the amount of substrate that is required is what I call as the Km value, also known as the Michaelis constant. Okay, now this Michaelis constant is a measure of how interested the substrate is on the enzyme. Okay, so to give you a very simple example, let's say your mother made paneer butter masala today, okay, and it's your favorite dish. Then what will happen? The moment I keep the substrate, my paneer butter masala is going to be over. Right? So very little substrate is required to reach half the maximum speed. So already the process has taken off in a great speed, right? So if the Km value is very less, it means that affinity is more. Okay? If the Km value is less, means affinity is more. But if Km value is very high, means what? Mother made upma. Upma is sitting in the table since morning. Okay, it is not cleared at all. Means more the substrate taken to reach half the maximum velocity, my affinity is low. So they are inversely proportional. Is that clear to you? So low Km means high affinity. High Km means low affinity. That's all. Okay. Now when you look at the line, okay, before going to the Lineweaver-Burk plot, there is an equation called as the Michaelis-Menten equation. Okay, so Michaelis-Menten equation states that my velocity at any given point of time, okay, or the reaction rate at any given point of time is equal to the Vmax divided by the substrate, multiplied by the substrate concentration. Okay, Vmax multiplied by the substrate concentration divided by the Km value plus the substrate concentration. Okay, so here this Km is nothing but that Michaelis constant that I spoke to you about. So if Km is low, affinity is high. If Km is high, affinity is low. So using the Michaelis-Menten equation, I can easily find out the rate of my reaction. Okay, now we have another equation in place called as the Lineweaver-Burk plot. Okay, now this Lineweaver-Burk plot is actually a double reciprocal for the kinetic analysis. Okay, so here what am I saying? I am going to take the I am going to change the Michaelis-Menten equation. So Michaelis-Menten equation is for the curve, correct? So I am going to linearize this Okay, so linearize this into a clear cut equation, okay, that states that V naught or the rate of the reaction is equal to, okay, any V is equal to the Km value divided by Vmax into S, okay, plus 1 by Vmax. Okay, this is the rate of this is the equation. So here what do we analyze? We analyze that when I linearize this particular equation, so first of all it will not be a straight line, something like this. Okay, here clearly my Vmax value or the 1 by Vmax value is going to be my y-intercept here. My slope is going to be Km by Vmax, and this graph in itself is 1 by S versus 1 by V graph. Okay, is that clear to you? So in this particular plot, what I find out using the linearized version of this Michaelis-Menten equation or the linearized form of the curve, I get to understand the different kinds of competition that is taking place. Okay, so if I look at the types of competition or the types of inhibition that can take place. Okay, the types of inhibition that can take place. First, I have competitive inhibition. So competitive inhibition means what? They fight for the active site. Okay. So my inhibitor is going to look just like the substrate. So the enzyme is going to think, okay, you're the substrate. It will go and bind to the inhibitor, but it's not the substrate. So my reaction cannot proceed. And as a result of which, obviously, my rate of the reaction will reduce. Okay. And that's it. That's about it. So my competitive inhibitor has already locked the active site. So further substrates cannot come and participate. Okay, that's the baseline. Okay, so here what happens is my Lineweaver-Burk plot looks something like, with the presence of a competitive inhibitor, looks something like this. Okay, so then what will happen? That intense. Okay, so if you look at it logically, the Km value will increase. Means now that the substrate cannot go and bind because the competitor has already occupied the active site, means my affinity has reduced. Okay, but the Vmax does not change. Okay, now in the case of non-competitive inhibition, okay, in the case of non-competitive inhibition, what happens is my inhibitor is going to bind to the enzyme, but it is not where the active site is there. It is somewhere else. Okay, in some other allosteric site, it will go and bind and it will bring about an inhibition. So in that case, what happens is my enzyme is here, okay, and this inhibitor will go and bind here. So as a result of which, what happen? It will change the conformation of my active site, and as a result of which the substrate now cannot bind, because I have changed the conformation by going and pressing somewhere else. The shape has changed. My substrate cannot come and bind. Okay, now in this kind of an inhibition, what happens? In this kind of an inhibition, my Km value is not affected. Okay, is not affected, or no change, I can say. Okay, so no change in my Km value. Okay, but the Vmax is reduced. Why? Because yes, my means my affinity has no change because the active site is not changing. Okay, or this thing is not changing. But what is happening? My total rate, like the maximum velocity I can go to, is reducing, because now that this shape has changed, I cannot bind anymore. So then my number of seats are reducing, means my number of means my rate of reaction also will reduce. So Vmax will also reduce in this case. Okay. Now there is another type of competition, another type of inhibition, which is called as an uncompetitive inhibition. So in uncompetitive inhibition, what happens is my entire enzyme substrate complex is locked. Okay, now if I lock my entire enzyme substrate complex in a way that no matter what you do, you cannot change into a product, then what will happen? Of course, my active site is already locked, so I cannot add more substrates to come and join my enzyme. So as it is, my Km value will reduce, affinity has reduced, and now this fellow is locked, so reaction also will not take place, so my Vmax also will be reduced. Okay, is that clear to you? I hope this concept is clear to you. So based on the Lineweaver-Burk plot, we can understand the kind of enzyme inhibition that is taking place. Okay, now if you look at the mechanism by which this enzyme catalysis happens, is very simple. You have an enzyme and then you have a substrate. So like a lock and key model, this goes and will bind to each other and form an enzyme substrate complex. Now this enzyme substrate complex, it's like a lock and key. So you open the key, so now it will form an enzyme product complex. Okay, now once the lock is opened, you can now remove the key, the lock is gone. In case you want, you can always put the key into another lock. Okay, so the enzyme and the product will separate, and now this enzyme can be used to bind with another substrate all over again. Okay, that is all is the mechanism. Now when you look at enzymes, there are some things called as the isozymes, okay, which are basically, it is the same kind of an enzyme, okay, but it's like different forms of the same enzyme. Okay, and this will catalyze the same reaction. Ma'am, why do I need the same fellow to act differently for the same reaction? For example, there is a enzyme known as lactate dehydrogenase. Okay, now for lactate dehydrogenase, there is one in the heart and one for the muscles, both of which are different because in different physiological contexts they will function differently. Is that clear to all of you? Now lastly, how are these enzymes regulated? Right, so for enzyme regulation, there are many mechanisms that you can take place. So first thing is using an effector, okay, or an effector through allosteric regulation. Okay, so in allosteric regulation, what is happening? The effector is going to bind to some other place which is not an active site, and it will enhance the rate of the reaction, enhance that enzyme to act. So it can enhance also, it can decrease also. So I can regulate the enzyme positively or negatively by using these kind of effectors. Okay, now apart from that, genetic regulation can be there. Okay, you know the operons that are there. When we are doing molecular biology, I'll teach you more about it. But using these operons, I can also do a gene level of regulation. Also, okay, I can use some proteolytic activation. Now what is proteolytic activation? I will have a simple molecule, okay, which is like a zymogen, which is inactive. Okay, in order to start the enzyme to work, I will have to convert this zymogen into an active form. So that way what will happen, I can regulate whether or not this fellow has to be converted and whether or not he is needed in this context. Okay, apart from that, there can be modifications like phosphorylation, okay, there can be acetylation and all that, which can modify the structure of an enzyme and regulate it accordingly. Okay, and there can also be a feedback inhibition. What is feedback inhibition? Now let's say I have an enzyme, I convert it into a product. Now once the product is formed, I don't want this reaction anymore. So this product will go and tell the enzyme that enough bro, I am done, so you can stop the process. Or if the product is very less and I need more product, again the product itself will go and it will tell the enzyme that no, I need more, so you start functioning. So like that, the product itself will determine whether or not the enzyme has to work or stop. Okay, so that kind of feedback inhibition also happens. Okay, now all of these notes I will be sharing in my Telegram channel. The description box has the link for it. So do join the Telegram channel so that you have all these notes in place. Additionally, like I said, we provide materials, and these are books curated from over 48 standard author textbooks. Okay, so these notes will be very crisp, okay, in the form of flow charts, pointers, okay, and you will get proper big volume textbooks that have detailed detailed explanation also. So not just for your CSIR, but also for your DBT, other exams, and also your future coursework, you can definitely utilize these materials. Additionally, we also have AI tools who is having a smart practice engine, and that can help you to enhance your practice, enhance your analysis, your understanding, and groom you according to what stage you are at, in such a way that you will be able to clear the exam. Okay, so I hope this session was useful to you. Definitely do check out my whatnexteducation.com website for details about our programs and to reach out to me for any kind of mentoring. Okay, so do subscribe the channel and stay tuned, because every single day I'm going to be helping you in a better journey to clear your CSIR exam. All the very best.